Hi, it's a pleasure for me to introduce Chinwe Tan for this afternoon's lecture. Chinwe graduated from the National University of Singapore in computer engineering and came to Melbourne University to do his master's degree in biomedical engineering. And I met Chinwe at the time that he was studying with Bruce Gardner and David Smith on simulating growth factor pathways. And they weren't sure which pathways they wanted to simulate, but after our discussions, Wnt simulation seemed to be feasible. And Chinwe has a real flair for MATLAB and all things software, and was able to start some computer simulations. But during that time, when we started to look at the effect of Wnt on colon biology and colon cancer, we started to think that maybe we could do simulations on a larger scale, namely the whole crypt. Crypt is, as Chinwe will tell you, 700 cells in a mouse and 2,000 cells in a human, but it's a very self-contained unit where you've got stem cells, mature cells, all happening all the time. And I've been in love with it for a long time, but Chinwe felt maybe if we got enough data that we could start to understand and predict the behavior of crypts. And whilst he started a lot of this from a computational point of view, he quickly realized that he had to gather some data, some quantitative data. And again, he showed this sort of very engineering approach to any problem. One, it had to be reliable, and two, the data had to be gathered in an accurate manner. And we're still doing that, but he's made a huge amount of progress in our understanding colonic crypts and the way they grow in vitro and in vivo. And I think you'll find an interesting talk, and I welcome you to the podium, Chinwe.
All right. Can you hear me? Hello? All right. Thank you, Tony, for that kind introduction. So I guess Tony has already put the basis on. I'm actually an engineer by training, so the past 5-6 years was quite an adventure for me, and it is something that I would like to share with you now for what we have been working on and discovered. So the title of my talk today is Wnt signaling and colon cancer in a computational systems approach. So I'll start with the overview of what I'm going to talk about. A few themes that we'll be covering includes colon cancer biology, Wnt signaling, systems modeling of intestinal crypts. Then I'll go around and just briefly run through some of the quantitative biology that we have been acquiring from a few years back, mainly in terms of quantitative measures of Wnt components in mammalian cells, quantitative measures of beta-catenin E-coherent in colon crypts, novel mechanism of colon crypt production that we came along, and we started growing colon organoids in the cultures, and then recently we have also started growing in vitro colon crypts in cultures. So we would like to tie it all back into a systems biology of the colon crypts, and let's see how I go with that. Some background. So colorectal cancer is the third most common cancer and the second leading cause of death in the Western world. Here I'm presenting you the incidence rates published in 2011, where I think the key thing to note here is that Australia and New Zealand are right up there at the top of the pile. So that is a substantial disease and a huge problem we need to address. So in my premise is we were trying to use different approaches, multidisciplinary approaches to try to look at this problem. Here, some biology. So the colon is part of the intestinal tract, where it's actually right at the last bit of the tract. So if you look at the surface of this, the colon itself is actually made up of thousands and thousands of infolds, which are infoldings of epithelial cells forming into the shape of a crypt. So that's what we usually call it, a colon crypt. This is just a layer of epithelial cells, and as Tony mentioned, it's actually made up of numerous different types of cells, from the stem cells, known to or supposed to be at the bottom, from proliferative cells, going and all the way up to differentiated cells. This is actually one of the colon is actually one of the most regenerative organs in the body, where actually the turnover is about within 48 to 72 hours. One thing interesting about this is that the cells started renewing at the bottom, and then it chucks up, moves up like a conveyor belt system. As it moves up, it starts to specialize and differentiate as it covers back up or the surface of the cells, of the crypts. So in 90% or more of these of colon adenomas, there's actually some part of mutation of the APC gene or some deregulation in this gene, which causes the regular shapes of these crypts to form various polyps, as we see. As you can compare here, where if you look at just the normal ones, they're just nice and cylindrical. The adenoma looks really pretty ugly in this sense. So after 10 to 15 years, if they acquire various oncogenic mutations along the way, become carcinogenic, a carcinoma will form. So I'm presenting here another slide of the sequence of events associated with the colorectal cancer development, from the normal epithelium all the way down to adenoma, carcinoma, to metastasis. And of course, right at the top, as I've mentioned earlier, are key mutations in these three particular proteins, namely axin, beta-catenin, and APC, which is called adenomatoous polyposis coli. So these are actually all key components of the Wnt signaling pathway, and that's one of the reasons why we are so interested in this. Wnt signaling is essential for intestinal homeostasis, and its deregulation has been found in over 90% of human colorectal cancer. So what's this pathway? It actually modulates the protein beta-catenin, which is a multi-functional protein. So I'll just run through some of the mechanism that has been known of this protein. There's still contentions about actually how all this works, but this is just a brief overview. So the basis is that two key scaffold protein, axin and APC, actually come together with the kinase GSK3 beta to form a degradation complex. This beta-catenin is recruited to this complex and is phosphorylated by GSK3 beta. This phosphorylated beta-catenin is then targeted by the beta-TRCP ubiquinate ligase, and is ubiquinated and subsequently targeted for degradation by the proteasome. So this is the off state where there's no Wnt signaling occurring. But when the Wnt signaling is switched on with the binding of the Wnt ligand to the freezed and LRP receptor, it actually causes beta-catenin degradation to be inhibited. One of the theory is that the degradation complex is actually recruited to the membrane, sequestered there, and is broken up. So without the degradation complex, beta-catenin is not phosphorylated, is not ubiquinated, and then thus it's not degraded. So that leads to stabilization of the proteins and nuclear translocation and subsequent gene activation and transcription. One of the functions of beta-catenin that has not been implicated in the Wnt signaling pathway is the E-coherent pathway. E-coherent, where beta-catenin is one of the key components of the cell-to-cell adhesion complex. In our lab, we actually believe that there's a huge relationship between E-coherent, cell adhesion, and beta-catenin in Wnt signaling. So Wnt stimulates actually the formation of various complex cellular structures like the colon crypt. Most of the literature will state that the Wnt activity is higher at the bottom of the crypt. And as Tony has already mentioned, that these crypts, in a sense, for modeling and all that, or modeling the structure, is actually one of the most ideal. It is just an epithelial sheet. It's like a cylinder, like a test tube. And it is very structured. It doesn't change much. So it should be reasonably easy to model this and to understand actually, well, is the wind activation really high at the bottom? But at this stage, most of the modeling literature that has come out has been mostly this form, where it's actually a 2D. So you're trying to infer a 2D system on a 3D context. But as I'll show you later, it's actually not that homogeneous all along the three-dimensional axis. So what we're trying to do is to get from a 2D to 3D. And there's also models that are starting to come up looking at building this whole crypt model from various components, where intracellular component, the mechanical component, positioning, where is it, how the cell adhesion is going, and also including the cell-cell cycle. But a lot of these are predominantly 2D data, and there's a lot of lacking in this kind of data at the moment. So what is lacking are quantitative 3D data to actually calibrate these models so that they can actually be predictive. One example is that for many years now, the wind model has been predominantly based on data from a xenopus oocyte. So this was in 2003. So this incompatible data is not helpful when you're actually trying to model them, as I will show later. So what we are actually trying to do now is try to build a model of the normal crypt to understand the biology of that, and then to also build a representative model of the adenomaceous crypt, and then to understand what is changed, what have changed, and for a better understanding of this, for better therapeutic targeting. So we thought, okay, let's start collecting data. So what's the first thing that we looked at was the basic wind signaling model. So my background is engineering, so we actually look at, okay, now we had tried to develop ODE models to try to model this. So the first thing is, like, let's get some data, and let's get some context data. So we started off saying, okay, let's look at, mammalian cells. So that's closer to our colon and human systems. So we looked at 293Ts, MDCK, which is dog canine cells, CACO2, SW480s, colon cancer cells, and then SW480 APC. So we started by doing Western blot, having recombinant proteins to actually use as standards to be able to quantify total protein per cell. However, what we really need for modeling is actually concentrations in terms of, for example, nanomolar. So we also want to look at it in 3 dimensions. So in this case, we started looking at, can we use techniques like imaging to actually quantify that for you? So to get concentrations from total protein, we need the volume. So we started looking at live cell imaging of, in this case, 293T cells, where we actually did 3-dimensional stacks of trypsinized cells, and we stained the cells with different markers to isolate the different compartments. So in that sense, we actually look at the protein concentrations for 5 of these proteins that I've mentioned earlier. And here's are the results. And you can see that there's a huge difference if you look at the different cell types. So these are the proteins that were quantified for different cell types. And if you look at it closer, you can see that there's a huge disparity between different cell types. And if we compare it back to where the xenopus system is, you can see that there are actually huge differences, particularly where in Lee et al.'s paper for the xenopus, they are stating that the axin levels are the limiting factors of the system, where it's at as low as 0.02 nanomolar. But in our case, where we're looking at different mammalian cells, is totally different. And in his case, he's claiming that APC is actually not the limiting factor, it's actually abundant, but on the contrary, actually very low in this system. So this sort of tells us that you have to be very context specific when you're actually modeling different systems, and you have to be conscious of which systems and what are the concentrations that you're actually using. So the other thing we are actually doing that, so that's a static point where you actually get a steady state concentrations. So it's very useful for modeling as a starting point. Then to actually get good dynamics, we have to have dynamic data. So now we start looking at classes of cells in 3D. So we want to look at spatial and temporal dynamic changes using confocal microscopy. In this case, we are looking at 293T cells, and because we are looking at different various time points, at different fixed time points, we're actually using the microspheres to normalize between the various time point samples. So what we did is, in the context of filling up some of the components we need for the modeling, is looking at extracellular Wnt factors like Wnt3a, and then look at the synthesis rate or degradation rate by using protein synthesis inhibitor, cyclohexamide. So as you can see here are the results of perturbating with cyclohexamide and Wnt3a. Bear in mind that this is a 2D section that we're looking at, but in actual fact, we're actually taking the whole three-dimensional stack of the whole cell layer. So you can see with cyclohexamide, what we're staying here is beta-catenin in green, and the beta-catenin level starts to drop within 4 hours, and the Wnt concentration starts to increase within 4 hours. So this is just to remind you that we are actually looking at it in a 3D context, where we're using the DAPI as the mask for the nucleus, and whatever else is actually the non-nuclear mask. So we also did validation to see how we compare between Westerns and confocal. And we did the actual same experiment, and with whole cell lysates for Wnt and cyclohexamide, compare them. And this just to orientate you, so the bold lines are actually results for the Westerns, and the dotted lines are results for the confocal. As you can see that the correlation is very good. So this gives us confidence that we are getting the right results. Furthermore, this was by using confocal microscopy, you can actually get a lot of localization data, which you will be hard to get if you're looking about doing Western blots. So now we have the first quantity measures of the key Wnt signaling proteins in mammalian cells. So this is very good for our modeling. Then in the context of the crypt, what we would like to know is where all these proteins are in terms of position, and how are the cell-cell adhesion functioning here. So it would be good if we can actually look at them in the crypts. So actually we can now isolate the crypts from the colon mucosa by actually separating them from the mucosa using chelation. So we apply the same idea where we apply 3D confocal microscopy to these isolated crypts, and to look in a simple sense just how these levels varies between the top and the bottom of the crypt in 5 different sections. So here are some of the results. So we are, again, I'll remind you, we're staying for DAPI, which marks the nucleus, beta-catenin, and E coherent. So this is one crypt, and we're looking at a 2D section through it. You can see that beta-catenin is quite homogeneous throughout, and does look slightly higher at the bottom. And you have the E coherent here, which there seems to be a bit of a lower level at the bottom. So looking at another crypt, going through the sections as we go up, you can see that this is an overlay of the 3 colors. You can see that there's actually quite a heterogeneous expression of this protein as you go through the stack. And in particular, you can see at this bottom here, as the slide comes back out, you can see that there's actually these few cells that's low, and then there's 2 cells that is actually very yellow. So what we discovered in this sense is that there's actually a certain population of cells where for the E coherent, there is actually a band of E coherent low cells at the bottom, always accompanied by 2, 3, or 4, a small cluster of cells that is actually E coherent high. And that sort of gives you a very heterogeneous expression at the bottom here, as you can see. So this is a 3D representation of it, and you can see that this bright cluster right at the bottom. But there's also this population of cells of crypts, sorry, that has no such heterogeneous expression at the bottom. So they are quite uniform throughout. So in terms of proportions, this is about 60% of the crypts that we have analyzed, which is about 30 of them. And 40% of the crypts are actually homogeneous. So we're not really sure exactly what's the critical point of this, but it does brings out something interesting. As we have done confocal microscopy, this is a three-dimensional analysis. We can also separate out the cytosol, membrane, and nuclear from the different compartments and then analyze them. So here is an intensity projection of beta-catenin for the cytosol, membrane, and the nucleus. So what I mean by that is that because it's a three-dimensional stack, we can't really display it as a 2D. So it has been actually compressed to show as intensity projections. Here is the quantitation for beta-catenin from membrane, cytosol, and the nucleus. There does appears to be a slight increase at the bottom of the crypt, and as a drop in the middle of the crypt. In terms of the E-coherent, you can see that it is clearly decreasing as we go down the bottom of the crypt. So that's all good. Then from there, now we can actually look at, we get the distribution of beta-catenin E-coherent in the crypts now. So one thing we did remember is that we can actually isolate these colon crypts from the colon now. So what about the morphology? Well, they are actually exactly the same as what we have seen in the mucosa. So what we can do now is to analyze all the crypts that we can isolate from the different sections of the colon, namely proximal, middle, and distal. So we try to check it out a bit. All right, and so we can get this, but that's not the only one we can get. We can get a lot of them. There's actually thousands and thousands of these colon crypts that we can actually isolate. And we're actually taking three-dimensional stacks through so we can actually quantify them. So this is a snapshot of me trying to use imaging software to actually start drawing all these dimensions. So you're drawing the length and the width. So quite a lot of work in terms of actually just drawing these lines. So a few group of us was really cross-eyed by the end of this whole episode. So this is the summary of what we have got. So just showing the proximal crypts, middle crypts, and the distal crypts for different age of mice. So 2.5, 2.3 weeks, 4 weeks, 7 weeks, and 40 weeks. You can see that, I guess one look, you can actually see that the proximal crypts are actually chunkier, shorter, and that the distal crypts are actually big and long. So we actually quantify it to actually show that in green, proximal is actually shorter on average. So that does tells you that if you just take the whole colon, there's actually different morphological difference between the start and the end. So here just to show you that we actually counted up to 2,000 crypts, and that really made us really cross-eyed after that. So having analyzed so many of these crypts, we tend to notice different things as it keeps coming through our view. So there's one thing that we did notice is that there's actually various different structures that actually turns out which is not what you usually see. So looking through the depth stack, which means you are going through the depth of the image, you can see that there are some of these that actually have sort of like a new crypt that's growing out. In this case, you can see that they're actually starting to grow out right near the bottom here. So by tracing through all these confocal images at different depth, we sort of have a different view of how crypts are starting to form. So in this case, it's actually an asymmetrical production mechanism, which we then start looking, okay, now categorize them, measure, and using averaging to say, okay, what's the basic deviation of this asymmetry is. And we find that about 90% of these crypts are actually asymmetrical. So typically, I think this will be missed by when we're doing 2D histology because the sections are too big and we're actually counting more than 2,000 crypts. So that's probably not really feasible for actually doing histology. And so we came up with this idea that there's actually a different forming mechanism, which is asymmetrical, where it actually goes through a stage where it's actually starting to split out near the bottom of the crypt and extend out from the side, and before it's become fully grown and then detach off. So this is a bit different from what has been in the literature for years now, where we're saying that it's actually a symmetrical crypt fission, where it actually splits into two equal crypts. So the other thing we also notice is that because we have data for various different ages of mice, that the amount of crypt fission or budding, and in this case we're calling it budding to just differentiate between asymmetrical and symmetrical budding, that the budding rate actually decreases with age. Well, in a way that's not surprising, but this also gives you a good indication for experiments, or that where's the point to cut, to stop, or to do the isolation. So one thing we also to note is that in yellow here, I presented what has been into the literature. So that is the budding rate that has been published. But we are actually getting significantly more because probably most of them actually miss the stage 2 here. So that's one of the advantages of using this three-dimensional imaging technique. So what then is this distribution of the key Wnt signaling protein in actually these different stages of crypts? We actually had a look back at what we have explained, and you can actually see at different stages, stage 1, stage 2, stage 3 of this crypt budding process, there's actually heterogeneity at the bottom. As you can see here, patches of green, right at the focal point of crypt budding, and you can see this is stage 1 where it's starting to bud out from the side. And initially when I started looking at this, I didn't really notice too much. So when we go back and have a look, this is something pretty significant. So there's something interesting that we would like to understand further, but the problem with this technique is we're actually looking into the mouse and taking the crypts out from the mouse. So to actually understand how this developed, we need to actually monitor the development in vitro. So that's what we started to look at. We need to investigate and follow the column crypt development in vitro and obtain the quantitative dynamics. So we need to go from isolated crypt to culture. So 5 years ago, the intestinal crypt culture has been established by the Sato Hans Cleaver's group, where they use 3D cultures and with isolated crypt that is embedded in matrigel. So you actually mix them up and let them grow with a certain set of culture medium. So but basically most of the things that they have got working are actually for small intestine. But we are interested in the colon, where all the cancers are. So, but the problem then was that the establishment of the colon crypt culture has been very difficult. So most of the ones that they have got growing are actually round cystic structures. Even up to 2 years ago, that's what they have got out. So what was the problem? We're not sure. One thing we did know is that all these are epithelial cells growing in matrigel. So what happens to all the other cell types that's actually growing beside it? So in vitro, they're actually having a lot of support cells, mesenchyme cells that's beside the crypt. So they might be secreting some factors that is helping this crypt to grow. So one of the candidates is the myofibroblast that is just beside the epithelial layer. And in fact, Magnus group has already shown that SOX9 solid cells can be co-cultured with embryonic intestinal myofibroblasts to actually cause colon crypts to grow. But they're taking more than 12 days, up to 24 days, before they start to bud. So what we did was, similarly, we actually created a stable cell line, a myofibroblast cell line called WEHI cells that is able to actually increase the efficiency of crypt formation in the colon culture from isolated crypt significantly more. So one thing to note is that from here on, because most of the notations for these cultures is small intestine, where they're calling organoids. So in this case, we're actually calling colonoids, where actually there's actually budding in the colon culture. So the culture system we're using here is with 24-well and trans-well insert, where you can see the reds are the isolated crypt, and it's actually embedded in Matrigel in the insert. The support cells, in this case the WEHI YH2 cells, is sitting at the bottom of the 24-well plate, and it's all embedded in medium. So there's actually no physical contact. So this sort of tells you that it's probably a soluble factor. And here are the results. So you can see for colon crypts only, that means there's no co-culture. They are actually not growing very well, and they actually die by day 10. The WEHI YH2 co-culture, you can see that they're actually growing and they start to bud. So that's good, and we can actually then start counting the colonosphere, which are the round ones with no buds, and the colonoids are the ones that has buds. So previously, most of the colon cultures have only colonosphere. So we can see that in this case, with the co-culture, we can significantly increase the colonoid count within 10 days. So the other thing that we noticed from this experiment was that previously most of the cultures had started from isolated crypts. So you actually isolate the crypt and then put them in culture. So of all the colonoids that actually form, there's actually a huge proportion of them that actually doesn't start from a whole crypt. So they actually start from fragments or single cells. We're not sure because they are quite hard to actually catch at this stage. But that tells you that there's actually different mechanisms at work here. So, so that's all good. But then there's a problem when we actually have Matrigel and different support cells inside. Our ultimate goal is trying to actually to stain for different markers and then to be able to do interventions, drug treatment. So we have a problem. So immunostaining of the colon crypts in situ with the Matrigel and support cells, that they present a huge physical barrier. And for biochemical interventions, support cells and condition media from support cells will introduce unknown factors. So we need to have a simpler system. So that's what we try to do. So from the embedded system, we actually change the system and say, instead of having them embedded, we're actually growing the crypts on top. So we're actually letting the crypt sit on top of the Matrigel in media. So this sort of works well. It is working. So you can see that we actually follow them up to 10 days, and they're actually budding well and pretty nicely. So, but the surprising thing that we got from this is that actually earlier in the culture, we actually see things like that. They actually cryptic colonoids. They actually look like crypts. So they actually are shaped like the crypt, and they look like crypts. So that was very surprising for us, and that's not unexpected. And usually you don't see that in embedded cultures. So with this system now, we can actually follow the crypt growth from day 0 all the way daily, day 6 to day 10. And then we take 3D stacks of it, so we actually can get most of the crypts in the well. So now we can actually follow them, as you can see, and see what happens to them, whether they merge or not, and then you can see them bud. So this is what we found. We have all the usual large body colonoids, as we call it, where there's a huge body, and the crypt starts to bud from it, or the crypt-shaped colonoid, or the in vitro crypts that actually start from a small sphere, and then they just round up and then form a crypt. But there's also other things that came out from this culture that's quite interesting. First of all, is actually multiple secondary buddings that seems to be occurring, as well as the dynamic nature of this system, where you can see that at this point, the bud starts from day 6, but then day 7 is not there anymore. And then day 8, it came out with gusto. So it seems that this system is very dynamic, and with this system, it actually allows it to grow. So then again, it goes back to the same question. So what's the distribution of the key Wnt signaling proteins in these in vitro crypts? So what does it look like here? And in the whole point is what we want to use this system was for immunostaining in situ. So can we do that? So that's what we tried to do. So we grow the crypts until day 10, or in between is fine. So in this case, day 10, and we stain for them. And voila, that's what we got. So we're actually very happy that we're actually getting these crypts, in vitro crypt that has a nice lumen opening at the top and a nice round bottom. We're also pretty happy to say that, okay, there's heterogeneous expression of this beta-catenin and E coherent at the bottom of the crypt. So that's all good. So here's another example of in vitro crypt where you can see the crypt shape, the heterogeneous expression at the bottom bits of it, and how much of a three-dimensional depth we're actually covering here. So without this actual depth here, we're actually missing quite a bit of different cell conditions here. So that, I think, is a huge emphasis of doing three-dimensional imaging. Here is an example of a large body colonoid where you can see here, this is actually a panel of 4 field views. So that's actually almost 200 microns big colonoids. And we're actually imaging this bit here. You can see again that they are actually growing, budding out in a very nice crypt shape. And from the side view, you can see that they are actually growing very well. Heterogeneous expression throughout, and we are still trying to learn what all this means. So comparing how the shape are looking, so you can see again with isolated crypt and in vitro crypt, we're actually having nice top open lumen opening and a round crypt bottom here. So we're comfortable with that. We also see how the crypts are growing. In culture, is it growing normally in culture? So they're actually developing normally, but what we did was to look at differentiation markers, namely chromogranin A and MARC2. So you can see that chromogranin A's have very few cells that is actually expressing that at the bottom of the crypts. And the mucin cells are actually all along the length of the crypts and in the lumen. So that's all good. So we are comfortable that they're actually growing well. So that seems to come up. We say, okay, now we're actually quite good. So now we have the capabilities to start actually building all these models in 3 dimensions. All right? People have already started doing that, but mostly in the small intestine, because that's what grows, and 2 dimension. So what we have to do now is, okay, now can we actually do the perturbation actually to build the Wnt signaling models and all the intracellular models for colon-specific cells? So that's one of the purposes that we will try to do. So we set up an experiment to look at perturbating small colonal spheres at day 2 with Wnt and cyclohexamide, as we have been talking about in terms of Wnt concentration and the synthesis rate, similar to what we did for the cell line. And this deprivation is a bit of a tricky situation because the colon culture needs Wnt to grow. So what we had to do was to put stabilizers in so that they are viable on day 2. So we still get nice round colonal spheres at day 2. And we just have to check that actually there's a drop in the beta-catenin stabilization of the, so that we know that the Wnt signaling has been dropped to the baseline. So that's all good. And similarly with the 3D cultures, we take 3D stacks going through the small colonal spheres. And here I'm presenting you an overlay of it, of the 3 colors, and 2D section of the beta-catenin slice, and again, an intensity projection. So here I'm presenting the data for the Wnt stimulation. Here are the images, representative images. So then, as you can see, the beta-catenin levels appears to increase as with time. And that's what the quantitation seems to show, that they actually increase about 20-30% with time. And with cyclohexamide, there's a slight drop and slight decrease, and as it slowly decreases at the end. So that's all good. And again, with this analysis, you can do the compartment analysis. So we separate out the nucleus, cytosol, and membrane compartment, see the individual increment for the different compartments, for the Wnt, and for the cyclohexamide. So there does seems to be a larger drop of beta-catenin level in the nucleus building cyclohexamide. Excuse me. So the other part of it, we can actually look at how the E-coherent is changing at the membrane throughout the stimulation. So it doesn't seem to have sort of significant change, but there's quite a huge variation in the E-coherent levels. And there's a slow drop of E-coherent with a cyclohexamide. So there's also other inventions of the colon crypt cells that we are actually working on with this new system, namely withdrawal, using cytokines, inhibitors, and then siRNA knockdown. So one of the withdrawal of the different factors we tried was to look at Nogin withdrawal. So Nogin is one of the BMP inhibitor, and it's one of the factors that we actually add into the culture. So by withdrawing it from day 2, we are actually getting a significant increase in the number of crypt-shaped colonoids in compared when you're actually using standard media. So that's one of the things that have came out from the analysis. And this is all still very preliminary. We've done some work with Toby Fies in cell signaling, looking at the effect of the JAK inhibitor and IL-11 on colon and small intestinal cultures. And we are also working with Dane Chisley in the system biology division, looking at knocking down endogenous genes like FILDA1, and we're seeing how the growth of these counts are with time. So that's all good. The other thing that we actually ultimately want to do is to be able to translate this to a disease model so that we can actually compare them. So in this case, we're actually working with Michael at the cell signaling division. We're getting some of his A33 CRE-RT APC flox mice, and to actually induce the APC truncation in vivo using injection of tamoxifen. And then we isolate the crypts from these mice after a week, and then put them in culture. And then we follow them for 7 to 10 days and see what happens. So preliminary, what we have got is, this is just the C57 normal black 6 mice. And this is what we got from the A33 positive A33 culture. So there are quite a few number of these that actually has elongated in vitro crypts, which is surprising. And we're going through the replicates at this moment. But this does tell you there's actually a morphological change, which is one of the key applications of this culture. Ultimately, what we want to do is to be able to grow adenoma. So recently, we got some of the colon adenoma from ANU, the systems biology division. And we're isolating, what we did was to isolate the crypts from the adenoma regions of the APC mean mouse colon, and then establish colon cultures from these crypts. So we are able to passage them, and up to now we are at passage 9 now, and that this is what we're getting. They are mainly predominantly cyst-like structures. So there's quite a huge significant difference, and we're trying to understand more of this. The thing about this is that now it's a very good opportunity for us to actually apply various drug treatments to this, and to actually get a lot of data and to try different drugs on this culture. So, so it all does seem that we're moving towards the systems biology of the colon crypts. So what we have done for a few years now is actually try to build Wnt signaling model from the cell lines, and we have started building it from the cell lines now with Wnt signaling data from the colon in terms of spatial distribution of various proteins and dynamics. We are hopeful that we will be able to actually incorporate all this data into an agent-based model with our collaborators over at Western Australia, David Smith and Bruce Gardner, to actually start building a lot of this predictive model. So at this stage, I think three-dimensional imaging, a lot of the microscopy techniques have come a long way, such that we are able now to be able to interrogate various different structures at various different levels, namely in the morphology level, cellular level, and the subcellular level. And with the advancement of the colon crypt cultures, I think in this case it is a good push to say that it is the right time to say that we are are now able to get a lot of this quantitative data in terms of the signaling, locality, dynamics, development from these cultures. So hopefully this will be able to be fed into the computational model to improve them from a 2D model to 3D and be predictive models of colon crypts. So to summarize what I have, so just to run through some of the parts that I have and what was previously there and what we have moved the system forward. So in terms of the Wnt pathway model, it was previously only in the Xenopus system. So now we have got it moving in nearer to the mammalian cell system. And then now we are looking at getting to a cell of context when in terms of looking at the colon crypt cells. So the beta-catenin distribution used to be 2D. Now we're looking at it in 3-dimensional immunofluorescence. And then we actually discover heterogeneous and homogeneous populations of these crypts. So in terms of colon production, the initial perception was it's a symmetrical fission only. So using this 3-dimensional imaging, we're able to identify asymmetrical budding and also heterogeneous points at focal points of budding. And from there, we actually started looking at small intestinal cultures, which was only on the small intestinal cultures, and to change it to a colon culture. And now we are able to do open colon cultures. And from there, we also observe a heterogeneity of colonoids. So now one of the things that we are actually looking at is to actually use live cell imaging, time-lapse imaging, to try to capture these various points of budding, to look at what are the particular markers that is causing this change. We also, with the ability now to do normal versus adenoma cultures, it is a good time to actually start filling in the gaps for the different components of the system's biologic model of the colon crypts, and to start building normal colon crypts models and adenoma colon crypt model, for which we will be able to understand the normal and the disease crypt biology, as well as better understanding of the mode of actions of this disease. So from that point, we will be able to more inform experiments and drug treatment, and with better experiments to act better for better targeting of these diseases. So, and what we're also doing then is using imaging to try to actually image a lot of these cultures to try to understand a lot more of the biology and where are the stem cells and where are some of the proliferative cells, differentiating cells. And I think this opportunity is very good at this stage. So with that, I would like to acknowledge Tony, and Tony for his mentorship and guidance, because without that, this will not be possible, bringing me from engineering to this great place here. And Yumiko, she's the main person, she's the instrumental person in this, where she did most of the experiments. And without this, without her help, without her hard work, this will not be possible. Kelvin is doing a lot of the co-culture system, and he's having his completion seminar in 2 weeks' time. So do keep a lookout on that, and we'll learn a bit more about what's in the cultures that are actually causing the crypts, the colonoids to bud. So in the system biology division, Oliver, Dane, and Anu for their help, and cell signaling division, Michael and Toby, and as well as the collaborators over at the University of Western Australia, David Smith and Bruce Gardner. And last but not least, I would like to acknowledge the funding bodies and HMRC. And with that, I thank you. Please.
Beautiful, very nice chemistry and lovely work. Just a quick question about I think if I understood correctly, there were ecoherent DD cells at the bottom of the crypts.
Yep.
And then you had juxtaposed sort of quite ecoherent, intense cells next to them. That looks very reminiscent of the architecture of the small intestine where the Paneth cells don't seem to express a lot of E-cadherin and the stem cells are very E-cadherin positive. Have you had a chance to have a look at those E-cadherin negative cells to see if they might in fact be the equivalent of the Paneth cell in the intestinal, in the small intestinal crypt?
Definitely, that's one of the things that we were also thinking of doing. It's actually a bit hard to now actually. One of the things about immunostaining is you have to get the right markers, and it takes a while to actually get the right staining. So we are thinking of doing that. But the other thing, of course, is what are the markers in the colon that actually marks the panel-like cells. So I think that's something of a bit of contention at the moment, and that's something we'll definitely be looking at.
Just with your in vitro cultures, with the embedded versus the non-embedded, do you think the non-embedded one is more like crypts because that's similar to the conditions in the colon for like not being embedded?
I'm not sure whether it's sorry, you're saying that it's more like in the colon, or?
Yeah, I mean, is it more like in the colon where the freer to sort of?
Well, I think that's probably one of the thing that we're thinking of. Personally, I think that as it's embedded, they actually have less room to maneuver, and most of the matrix cells have various basement membrane factors inside also. But then if it's all around it, it doesn't know which side to go and which direction to go. So that's why probably we're seeing a lot more dynamics in the open culture where they're actually trying to probe which area to go. So that probably also emphasizes a bit about the niche that it is at, and that you need to be aware of the different niche. And one of the things I didn't mention was that actually we're also thinking of looking at the different type of environmental gradients and all that coming from this system. So that's some of the things that we looked at. But definitely that's something we're interested to understand a bit more.
Jason?
Do you have an idea of the sort of rate of budding in vivo, and do these whole crypts get turned over in the colon? Do you know what I mean? Like if they're separating and forming new crypts,
Yep.
is that happening all the time? And where are the old crypts going? And are they turned over?
So in vivo, what we know was, well, most of the snapshots that we have done to see that they are actually the rate of budding that we're getting. But in culture, it is actually, at this stage, we're not getting that fast a turnover. And usually, these cultures finishes 10 to 14 days unless you passage them. So renewal-wise, we're not really sure at this moment because we don't really do a lot of the renewal cultures at this stage.
Yeah.
I'm just wondering if, maybe not with the colon stuff you've done here, but with the organoids from the small intestine, has it ever tried to grow pathogens in them?
You mean us or?
Well, I'm sure you have, but I'm just wondering if you knew of anyone that had used them to look at pathogen interaction.
Not that I know of, but I think there probably will be, yeah. I think Toby might know a bit more. I think.
Yeah.
I'm just wondering, does your in vitro culture has the same stem cell, a similar stem cell in the bottom of the crypt as compared to the in vivo culture?
That's what we'll be trying to find out. But again, we need to be able to mark the stem cells.
Yeah, and also, what is your source of cells to the organoids? Like what is your first?
Fragments.
Huh?
So crypt fragments.
Okay.
In the 3D culture, you seem to place quite a lot of emphasis on the levels of beta-catenin, and I wonder if you're making any assumptions about the state of quick pathway activation. So are you looking at the phosphorylated beta-catenin, or are you looking where it's localized?
You're right, you say that we are looking at actually the total beta-catenin at the moment. So then to look at, definitely we need to look at a bit on the phosphor side, but it also depends on the levels of phospho-beta-catenin you can detect in using immunofluorescence. So that's always one of the issues. And again, we need to work on that. Yeah, but that's one thing.
And can you see it in the nucleus? Can you distinguish where you get nuclear beta-catenin versus non-nuclear beta-catenin?
In this case, yes, we can actually see nuclear versus non-nuclear, but whether it's phosphorylated or not, we don't know.
Following on from that, if you have a look at nuclear beta-catenin levels, does it look equally as non-homogeneous as when you look at total beta-catenin? Because that's a more sensitive marker for active Wnt signal.
At the moment, it seems to be quite homogeneous across the board in terms of the whole near the bottom. But I haven't really gone in and actually picked out each single one. But there is, we're just starting to look at that for the manuscript where we're actually seeing some, I haven't presented here, but there's some cells where you have ecoherent high, low because of different levels. And then there's actually a slight difference in the nuclear beta-catenin in that sense between, for example, ecoherent high beta-catenin low cells on membrane versus ecoherent high and beta-catenin high cells on membrane. So there is some differences, but as for whether there's specific reasons for that, that's something we're looking at.
Last question.
Have you looked at from where budding occurs? Like, is it more from the middle region or the bottom region or top? Is there a particular region where there's more budding?
You mean the isolated crypts or
The isolated crypts.
mostly from between the middle and the bottom. So the exact spot, we're not too fast about that, but most of the ones that we've seen is starting from right near the bottom. That's why we had that picture. So it's not at the bottom. It doesn't split out mostly like that, but it is just off bottom, or maybe +3, +4 position or something. Yeah.
So, Chin Wei, thank you very much.
Thank you.